So I think to save time, we're just going to introduce ourselves. My name is John Dahl. I'm in the biology department. I teach microbiology classes. I'm particularly interested in infectious disease, so for me it was a pretty easy jump to think about zombies. Tonight, we're not going to think of zombies as supernatural creatures. We're actually going to think of them as really putrid flesh and tainted blood organisms that obey all the laws of physics, albeit they tend to be a little mobily challenged. Not all of them. Some are shufflers, some are runners. In general, zombies tend to be maybe not a few sandwiches short of a picnic, shall we say. They kind of tend to, they tend to let themselves go a bit. They're not the best people to have at a party because they can't really hold a conversation. They have an incredible case of the munchies for us, and they're pretty messy, particularly in holding down their blood. They tend to spit it up a bit. And so when you look at actual diseases, you see a lot of these symptoms. And what I would like to point out is that all the diseases that are listed here do have an infectious basis or can have an infectious basis. We're going to talk about two of them tonight. Myself, I'm going to address Ebola, and then the next speaker is going to talk about Kuru. So there's precedents for this in zombie films. I just mentioned two. 28 days later, an infectious agent there was a virus called the rage virus. It was spread in blood, very fast acting, and one of the symptoms from this virus was almost immediate transition into a zombie and spitting up large quantities of blood. Now that's different from the germ in Zombieland. This is a quote from the first movie. Mad cow became mad human, became mad zombie. So this implies maybe a prion-based infectious agent that might be slower in infecting and working its way through a human body. And our second speaker will address that. So tonight, in the next 10 minutes, I want to talk to you about the 2014 Ebola outbreak in West Africa. And I'll try and set the stage for this. So in the spring of 2014, really the entire world watched with concern, with sympathy, with appalling fascination of what was going on in terms of Ebola in West Africa. And this changed to fear in the summer of 2014 when infected healthcare workers were being shipped back to the United States and Western Europe for treatment. And there was a concern that the virus was so virulent that it was going to escape from the hospitals and infect Western countries. And so the threat kind of changed in people's perception of it. And people started to think of Ebola now as, could this be the next big one? I mean, by that, would this be something like the Black Death in medieval Europe? Or could it be something like the Spanish flu that killed upwards of 50 million people? I mean, people started to view this almost as if it was a biblical plague of Egypt, where some angel of death was grabbing the firstborn of every family. But we know then, it was a biological entity. It's a virus, which basically is an obligate intracellular parasite. It must find a host cell to replicate in, but it is living and it must obey the laws of natural selection. And I want to impress upon you the story of the 2014 epidemic is very much a story of evolution. So what does Ebola virus do? Well, essentially it's an infection of the circulatory system. Both the virus and your own immune response breaks down circulatory tissue, which leads to a massive amount of hemorrhaging. And so blood builds up and it basically tries to find a way out of the body. Blood can escape from the skin, from the mouth, from the eyes, and then huge quantities of Ebola-infected blood can leave the body by diarrhea and also by vomiting. It's a very fast-acting virus. Usually it enters the body either by a break in the skin or it comes in through the mouth. We know now that Ebola can also be transmitted by sexual intercourse. And if a person has been infected, the early symptom is pretty general. It's flu-like, progressing to a very bad headache, and then the massive diarrhea, and essentially a complete collapse of the circulatory system and a multiple organ failure. It has a 70% mortality rate, and generally the people who survive this either were given support medical treatment or they had a very strong immune system. But this 21 days, this is generally considered the waiting period. If you suspect you were exposed to the virus, you really had to be quarantined for that length of period before you were allowed back out into the general community. So there's a couple oddities about the 2014 epidemic. First of all, if you look at this timeline, what you see here is that every previous Ebola outbreak happened in Central Africa. Now that may not seem like a big deal, but Central Africa is 2,000 miles from West Africa. So it was a great mystery as to how it even showed up in West Africa. Well, we know that Ebola is a zoonotic infection, meaning that it is an infection that comes from animals. We're not sure which animal it's hiding in the jungle between these sporadic epidemics. We suspect bats, and bats certainly are mobile, and so the mobility is probably what led to the transmission over such a great distance. Another question about the Ebola outbreak of 2014, why was it so bad? So all infectious epidemics really kind of can be lumped into two categories. You either have what's called the common source, sorry, I'm getting used to my buttons here. The common source, which is, it's really pretty rapid, and this is usually due to some infection that comes from either a food source or a water source. That's different from an infection that's transmitted from person to person. This can lead to propagated epidemics. These usually have a set pattern where they crest, and oftentimes this plateauing can be due to breaking transmission between people. Maybe you're immunizing people. Maybe you come up with a medication. But eventually, you sort of get a grip on the epidemic. That's not what was happening in 2014. In the fall of 2014, what really freaked people out was that the epidemic, according to the Centers for Disease Control, went into exponential mode, which meant that it showed no signs at all of plateauing. And there were mathematical models from the CDC that estimated that if nothing was done to curtail the infection, by January, upwards of 1.4 million people would become infected. That's not what happened. 28,000 people were infected, but there was a great concern that this was going to go global. I think most of us living in Minnesota, we really don't appreciate how close we came to dodging the Ebola bullet. And I want to explain that what made this epidemic different was that, well, first of all, Ebola and all the previous epidemics in Central Africa were in rural settings. In 2014, it was now approaching into large urban areas, large urban areas in Liberia, Guinea, and Sierra Leone, and it was actually approaching cities. And that was a great concern because there would be a bigger pool for the virus. But these were cities that did not have international travel. There were airports, but they were local. That changed on July 20th, 2014, when an American Liberian lawyer named Patrick Sawyer caught an airplane from Monrovia, Liberia, to Lagos, Nigeria. And he ignored the fever that he had, and basically he was bringing Ebola with him. Now, Lagos is a city of 20 million people, very densely packed together. Some slums in Lagos have 50,000 people packaged into a 1 kilometer square region. And more importantly, Lagos definitely had an international airport with flights that were leaving daily. And in fact, Patrick Sawyer had a ticket to Minneapolis 3 days later. So disaster was avoided by 3 lucky breaks. First of all, he caught a plane. If he had taken a bus, he would have infected many more people. And more importantly, because he caught the flight, he had to pass through immigration. And the custom officers could clearly tell that he was sick, and they ordered him to a hospital. So that's lucky break number 1. Lucky break number 2 was that at the time in Nigeria, the country's entire healthcare system was on strike. So all the hospitals were shut down. So he was taken to a small private hospital where there was a physician, I'll hopefully pronounce her name right, it's Dr. Adadio, who immediately suspected Ebola. She had his blood drawn and almost immediately had a positive diagnosis for Ebola. Now, Patrick Sawyer did not accept this. He literally pulled the IV from his arm. He sprayed blood in the clinic. He really wanted to get on that plane and go to Minneapolis. He was ordered to stay there, and in fact, 2 days later, he eventually died from Ebola. Lucky break number 3 is that this event is happening in Nigeria, which probably has the most advanced healthcare system in Africa, and they had prepared for something like this. So immediately they sent out 40 epidemiologists and 140 healthcare workers. They tried to track down every individual, and they found 72 people who had direct contact with Patrick Sawyer between the airport and the hospital. And eventually 526 people were quarantined for this 20-day period to see who actually was going to develop the disease. Of that 526, 20 did come down with Ebola, and 11 died, including the doctor who made the original diagnosis. But what's important to emphasize is that by the end of October 2014, the epidemic was under control in Nigeria. In fact, it was considered gone from that country. So there's a scanning electron micrograph of Ebola right there. It's a filamentous filiovirus member. It's similar to Marburg, which is also a hemorrhagic fever. It's a pretty simple monster. It basically has a single RNA chromosome with 7 different proteins wrapped around it. The most significant protein is probably the one on the surface. This is called a glycoprotein. This is what's known as a viral spike, and that spike is what allows it to bind to a host cell. Remember, viruses have to bind to you and enter into your cells to replicate. And almost immediately with the epidemic, what happened was they started to take clinical isolates and do genomic sequencing. They wanted to see, where did this virus come from and how fast was it evolving? And I want to impress upon you how dangerous work this was. So this is a phylogenetic tree. Basically, it's an evolutionary tree. We have a good idea where the first transmission event happened. The first 78 isolates that were sequenced and started to build this tree, that paper came out in Science magazine. It had 15 authors. Of those 15 authors, 5 had died of Ebola before that paper was actually published. This is an incredible amount of work. This is over 1,000 clinical isolates that were sequenced. And when you look at this data, one thing that really pops out is that early on, there's the appearance of a mutation. And there's real strong positive selective pressure for this because this becomes the dominant form of the virus, this mutant. In fact, 90% of the 28,000 people who came down with Ebola had this strain, had this mutant form of it. So what the heck is up with this mutant? Well, it turns out that it was a single base pair substitution that led to one switch of an amino acid. So it was an amino acid switch from alanine to valine on the glycoprotein. This was probably allowing the virus to bind to us better. Now, the way that you prove that is you do the genetics. Usually with genetics is you knock out a gene and you ask, is the mutant, has it lost some function? In this case, it was thought the mutant gave it some function. So this is forward genetics. There's a lot of data here. I just want to show that what they were doing was they were trying to reconstitute the virus into something called a viral-like particle. This was not Ebola, so it wasn't going to kill people working with it, but it could show that by playing around with this mutant, they wanted to see, did it increase in infectivity? The assay was not to look how much virus was produced. They had a reporter system, so the more cells infected with luciferase, the more light it gives off. So they're actually monitoring light. I just want you to see this one piece of data right down here. When you actually had a viral-like protein expressing this mutant glycoprotein, this mutant spike, you had a 3.5-fold increase in infectivity. This is an image that shows cells in tissue culture, and that little spaghetti filaments there, that's the Ebola attaching to a cell. So it's pretty clear now that what happened in the fall of 2014 was an evolutionary event, and I guess I would say that if evolution can create a monster, it can also tame one. And by that I mean this strain was so virulent that it basically burned through the human population so quickly that there was no fuel left over. And we now believe that this strain of the virus is actually extinct. So it made an evolutionary gamble. It was super virulent, but it now is thought to be gone. I would say that what eventually brought the epidemic under control was just dumb luck that the virus didn't mutate further. An international effort that was spearheaded by leaders like Barack Obama. And I think the third reason the epidemic was brought under control is basically just a lot of brave women and men who refused to leave the bedsides of sick and dying people. And I want to tell you a story about one of those individuals. But in doing that, I'm going to bring back one more symptom about zombieism. So we talked about the blood projection from zombies. I want to talk about the eyes of a zombie. And if you look at this, I think we can all agree that when you look a zombie in the face, they look a little odd. There's something just not right about the eyes. They kind of glass over, they might change color, maybe there's a little bit of bleeding. So let me finish by telling you the story of Dr. Ian Crozier. Dr. Ian Crozier was a 43-year-old American healthcare worker who was working in West Africa and became infected with Ebola. Now remember, this virus has a 70% mortality rate. He was rushed back to Atlanta, and great effort was made to save his life. To do this, they brought in a British nurse who herself had had Ebola, recovered from it. She provided blood transfusions. It was hoped that her antibodies would save Dr. Crozier. He was also given an experimental drug called TKM Ebola. And basically, to make a long story short, he had multiple organ failures. He started going into seizures. He was unconscious for 40 days. And during that 40 days, every day, 8 to 10 liters of diarrhea is leaving his body that's full of the virus. Eventually, he did recover. He was 30 pounds lighter. He had to relearn how to walk. But a really unexpected symptom was that the color of his left eye had changed from its normal blue to green. And when they sampled the fluid in this, they found that, in fact, his left eye was loaded with the Ebola virus. They were worried that he would lose vision. Eventually, he was able to clear the virus from his eye, and the color converted back from green to blue. So another sort of real-life symptom that can kind of mimic what we see in zombies. So thank you very much. I'm excited to sit down now and listen to whatever the next person has to say, which is going to be awesome. much.